So last week we talked about cell structure and function in prokaryotes and eukaryotes. So today we're going to start looking at membrane structure and function, particularly in eukaryotic cells. So let's get started with that. So let's make sure that we have our terminology correct. So this is just some common terminology when you're talking about cells. And for example, let's say this blue oval is our cell. Anything outside of it we would call our extracellular space or our extracellular environment. Everything within the blue oval would be the intracellular space or intracellular environment, which is where our cytoplasm, also called cytosol, is. And what divides the extracellular space from the intracellular space is our plasma membrane. So there are four main parts to biological membranes. There's the phospholipid bilayer, proteins, cholesterol, and carbohydrates. Now, for the most part, we're going to be talking about the plasma membrane, but keep in mind that within our eukaryotic cell, we have several membranes because every organelle has a membrane that separates it from the cytosol. But for the most part, we are going to be talking about the plasma membrane. Okay, so when we take a look at our phospholipids, which if you'll remember from our organic chemistry conversation, we have the polar heads and the fatty acid tails that are nonpolar. And so when we look at those, they tend to self-aggregate in such a way that they compose two layers. And so that is why we call this a phospholipid bilayer. And so the phospholipid bilayer means that each layer is composed of a polar head and a nonpolar tail. The nonpolar tails are hydrophobic, which means they do not like to interact with water. So in order to escape the water, they point towards each other. And so that's that inner layer that has the hydrophobic tails. Now, the hydrophilic heads, or the polar heads, face the extracellular space, and they also face the intracellular space. So in order for them to interact with water, the extracellular space would contain water, and so one of those phospholipid one of the layers of the phospholipids would have its polar head towards the outside, and then the other layer would have their polar heads facing towards the cytosol. In order to describe the phospholipid bilayer, we have what's called the fluid mosaic model. Now, the fluid portion of that is because the phospholipids move around within their layer. So there can be lateral movement or even rotational movement of each of the phospholipids. Very rarely they will flip from one layer to the other, but that's difficult for them to do because they have to cross that either the layer of water, and so those tails would be stuck in the water and be very unhappy. So that doesn't happen very often. But within the layer itself, there's a lot of movement, so it kind of looks fluid. The mosaic portion is because there are other structures that are embedded in the bilayer. And we see this when we do what's called freeze fracture electron microscopy. So this image up here on the top left is taken using a transmission electron microscope. So the electrons are sent through our lipid bilayer, and you can see the membrane right there. For freeze fracture electron microscopy, it really is exactly what you would expect. It is the freezing of that membrane, and then as we crack it open, we get to see the two different faces of that bilayer. So we have the cytosolic leaf and the extracellular leaf, and those are the two as we would denote how that basically is the barrier between the cytosol and the extracellular environment. And so when we see that cracked open, we can see, if you see like these green spheres that is showing you that there are things that are embedded within the membrane other than just the phospholipids. And that is the list of the other three things that we talked about that are in our membrane. Okay, so this is just a model of our membrane. So we have the phospholipid bilayer, and then one of the things that we find embedded in that membrane is proteins. We see that from the freeze fracture electron microscopy. And there are two types of proteins that we find within our membrane, and the first of those is what we call a transmembrane protein. So if you remember your Latin a little bit, trans means across. So this protein crosses both bilayers, both of the phospholipid layers. So it has a portion that touches the extracellular region and another portion that touches the cytosol. So this is what one of those integral proteins or transmembrane proteins would look like. So if you'll remember when we talked about the proteins, their primary structure is a string of amino acids. So that's what we're looking at here. And so if we take, it would have portions here that are hydrophilic, so they would stick out in the water. And then portions that are hydrophobic that get folded into that inner layer, so this would be near the fatty acids of our phospholipid bilayer. And then other hydrophilic portions that face the inside of the cell, so at the intracellular space. And you can see how it winds back and forth. And this is what we would call an integral protein, also called a transmembrane protein. So the other type of protein that we have within our membrane is what we call a peripheral protein. And so that just means that it is only on one side of the membrane or the other. So it doesn't go through the entire membrane. This one, the example here, looks like it's on the intracellular side. It can also be on the extracellular side as well, but not in both. Okay, let's talk about the functions of these proteins that we have within the membrane. To start with, let's look at A and B, the channel protein and the carrier protein. These essentially act as tunnels to allow things to come either into the cell or out of the cell. And so obviously these are definitely going to have to be integral proteins or transmembrane proteins. They have to go all the way across. C is a cell recognition protein. And this basically just acts in order to ID the cell. Kind of like a fingerprint of a cell. Different cells have different cell recognition proteins. And then D is our receptor protein, and this transfers information. We're going to talk more about this on our next lecture when we talk about cell-to-cell signaling. How does one cell talk to another? It transfers information via these receptor proteins. And then E is our enzyme proteins, and we talked a little bit about those. And so basically these just speed up chemical reactions within the cell. So when we were talking about channel proteins and carrier proteins, and we mentioned that they are tunnels, basically a tunnel between the inside and outside of the cell, the difference between those is that the channel protein itself literally pretty much is a tunnel. If you see here, a solute can go straight from extracellular space to the intracellular space. It can also work in reverse. Our carrier proteins, however, tend to have a binding site. So the solute would come in, and once it binds, it changes the shape of the protein such that, here, let me grab my pen real quickly. So when it binds right here, it's going to change the shape of the protein such that the solute can then be emptied out. And so that's how the carrier protein works. So you can see why these two need to be transmembrane proteins and be able to cross both layers of the phospholipid bilayer. So the third component to our membrane is cholesterol. And if you'll remember from our last conversation, cholesterol is a steroid, and it's composed of 4 fused hydrocarbon rings along with a carbon chain. And so if you look at our model here, you'll notice that cholesterol is actually kind of small, a small molecule compared to the others, but it plays a significant role. So our cholesterol steroid has what we call a hydroxyl group, which is an OH, and that interacts with water really well, as does the polar head of our phospholipid bilayer. So that interacts with the water layers, but cholesterol typically, the majority of its molecules sticks within the hydrophobic areas, and it acts to stiffen the membrane itself. So there's the more fluid portions of the hydrophobic tails, but when you add a cholesterol to it, it stiffens that area. So cholesterol is found in animal cell membranes, and when you ingest fatty foods, that is one of the ways that we get cholesterol. The other way is that it can be synthesized in the body, particularly in the liver. So cholesterol functions to stabilize and strengthen the membranes. So for instance, if you look at this image here on the left, you might notice that some of the hydrophobic tails are just going all wonky and wherever they want to go. And then the cholesterol can come in and stabilize those tails to make sure that they're orderly. And so that is one of the main functions of cholesterol in the cell membrane. So the fourth component of our membranes are carbohydrates, and these function as flags or ID tags that identify a cell. Essentially, each cell has a fingerprint. It's unique, and most of the reason for that are the various carbohydrates that are linked to various parts of the membrane. So we find those carbohydrates in 2 different forms. One is what we call a glycoprotein, and so that essentially means that one of the proteins in the membrane has a carbohydrate chain attached, and so we can see that here on the left. This one is an integral protein with a carbohydrate chain attached. That's a glycoprotein. The other role for carbohydrates is as a glycolipid. So that means lipids in the membrane that again have a carbohydrate chain attached. So here is our lipid, and here's our carbohydrate chain. And based on the variation of that carbohydrate chain, and then which protein it's attached to, which lipid it's attached to, that cell will have a unique set of those carbohydrates, and that is the fingerprint of that particular cell. So a really good example of how these carbohydrates help to identify cells is with red blood cells. So by now you probably know what blood type you are. I am blood type A, and so red blood cells have glycoproteins. So in other words, in their plasma membrane they have proteins that have a carbohydrate chain attached, and that helps to identify them as A, B, or O based on what type of carbohydrate is attached to the various proteins. So, like I said, they do serve a particular purpose in identifying cells. So now that we've talked about what the membrane is made up of, let's talk about the function of our membranes. So this kind of puts it together nicely. What we call our cell membranes selectively permeable, which means they allow some things to come through but not others. So our cell membrane allows the cell to maintain its internal composition separate from the extracellular spaces. And it does this by allowing small uncharged molecules to diffuse through the membrane, but at the same time it it does not allow other molecules such as large polar molecules or any charged molecules such as ions. Those don't cross by themselves. Now, those things can come through selectively when we use some of those proteins that we talked about. So transmembrane proteins that are there are pumps, channels, carrier proteins, and we're going to go into a little bit more of that in our next lecture, but they are selective. And then the transmembrane proteins contain multiple membranes spanning regions. So like we saw, the amino acids goes in and out and in and out, goes across several different things. The function of all of these proteins then is to be selective about what it allows in and out of the cell. So let's divide this up a little bit, because we have our plasma membrane or our cell membrane, but then we have lots of membranes within the cell, and we call those our endomembrane system, endo being within, so within the cell. So our plasma membrane defines the cell boundary. It regulates the transport like we talked about with selective permeability. It allows for cell-to-cell communication, and it has the localization of certain structures within its membrane. The membranes within the cell, so think about like the nuclear envelope is one, the endoplasmic reticulum. All of our organelles are enclosed within a membrane. So our chloroplasts within our plant cells, things like that. Our mitochondria, for instance. And so the role of the membranes within the cell, or our endomembranes, again, compartmentalization, just like the plasma membrane, that allows each organelle to have a suitable environment, such as the right pH, certain molecules that may be necessary for the functions of that particular organelle can be sequestered into that particular organelle because of its membrane. And then it also provides surface area and specific enzymes that the various organelles need to accomplish their goals. So what I mean by that, and we're going to go into a greater detail with this later, is, for instance, our chloroplast, for photosynthesis, it has specific proteins that allow it to capture light, and those are only embedded in the chloroplast membrane. So it's very specific to that organelle, and so the membranes of various organelles do play a specific function. So if you'll allow me to, I would like to go back to the membrane proteins for a minute. We've talked a little bit about what the membranes are composed of, and one of those categories was the proteins. But when we were going through them, I purposely skipped over junction proteins. And the reason for that is because now that we've talked about the membranes of the cell and focus mostly on the plasma membrane, now we can talk about how cells interact with one another and how cells create something on the outside called the extracellular matrix, and we'll talk about that in just a second. So I just briefly want to mention these junction proteins, and we find these particularly in animal cells. And so what they are is transmembrane proteins of one cell and then another transmembrane protein of another cell that chemically bond together, essentially linking those two cells together. And it's used for a variety of purposes. So I want to talk a little bit about the connections from cell to cell. So let me just briefly dive into plant cells, and then we'll come back to animal cells. So let's talk for a brief moment about cell junctions. And what I mean by that is places where the plasma membrane of adjacent cells interact. To start with, let's look at our example in plant cells, which is on the left side of your screen. Plant cells have what we call plasmodesmata, and plasmodesmata are essentially channels between two cells that are lined with plasma membrane. So it allows for quickly sharing molecules between cells. Now, this is especially important in plant cells. I mentioned before that there are some cells called sieve tube elements, and those sieve tube elements don't have a nucleus at all. They are involved in the transport of sugars up and down the plant. So they act almost like a straw. And what they always have with them is what's called a companion cell, and that companion cell has a nucleus, and it regulates the metabolism of both the sieve tube element and itself. So these plasmodesmata become very important in that relationship because genetic material as well as metabolic molecules can pass back and forth between those plasmodesmata. Now, in animal cells, we have just a few that we're just going to briefly touch on. And the first is an adhesion junction. And so what that looks like is, if you can look here, there are these plaques that form, and that's part of the cytoplasm. And then lots of densely packed areas of intermediate filaments of the cytoskeleton. And then there are proteins across here to help them adhere to one another. And so we see these often in muscles, for instance, or in skin, things that stretch a lot. These adhesion proteins allow for that stretching to occur, but those cells to still stay together. The second one is a tight junction, and it's really a type of adhesion junction, but it actually keeps the cells even tighter together. And the way that works is the proteins basically put the plasma membranes of two cells together in a way that almost works like a Ziploc bag. It keeps them nice and tight. And so that's important in areas where you really don't want to let certain substances move across a tissue layer, for instance. One example might be in your kidneys where we have urine being created. You don't want that to move across your kidney barrier. So we have a lot of tight junctions in the kidney. Our other junction in animal cells is called a gap junction. And basically that is allowing plasma membranes of adjacent cells to be connected with these channels. And so a channel is usually lined with 6 membrane proteins, and it allows typically for ions to flow back and forth between those cells. And that's especially important when you have a lot of cells that need to work in conjunction with each other. And so, for instance, the heart muscle depends on ions in order to contract, and you don't want all your heart muscles to be contracting at different points of time. So to allow for them to contract at the same time, these gap junctions are present in those particular cells. So those are just some examples of how plasma membranes interact with one another. On the outside of animal cells is what we call the extracellular matrix, and that's basically just a three-dimensional network of macromolecules. So a lot of animal cells will excrete various macromolecules, mostly proteins, but collagen enzymes, glycoproteins, that provide extra structural support to the surrounding cells and are also involved in communications from cell to cell. So that's what we find in animal cells. In plant cells, we have a cell wall. So here we have our plant cells, and to the right you can see each one of these blocked off here. That is a plant cell. So in our plant cells, outside of the cell membrane, we have a plant cell wall. So in the image to the left, here is our cell membrane, that phospholipid bilayer. And then what composes the cell wall are these long cellulose fibers. Remember we talked about during our organic chemistry that those long carbohydrate chains of glucose makes up cellulose. And we said that was the most abundant of our carbohydrates, and so here we are. And so the cell wall is not actually an impenetrable wall like we think of. It's more like a mesh layer of cellulose, what we call hemicellulose, which has different monomers than our cellulose, pectin molecules, things like that. So there's water and small molecules that flow within this mesh of cell wall. Just to give you an idea what this is, this middle lamella, that's actually the name of the space between the cell walls of two different plant cells. Okay, so we started out today, our goal was to talk about membrane function and structure, and we did, but I expanded that a little bit just so that you know how cells interact with each other and what's right outside of the cell membrane, because oftentimes we talk about the cytoplasm, so we know what's inside the cell membrane, but I wanted you to have an idea of what is outside of it also, so you understand the environment that it works within. Okay, so next time we talk, we're going to talk in detail about transport across those membranes and how that works and how it's regulated.